Homework 3. I intended for the bootstrap to be optional. If you didn't want to do the bootstrap, you could use the t-test to get the p-values. So you could either use the bootstrap to do the p-values or use the t-test to get the p-values, is what I intended. If you didn't do it at all, that happened at least once I know of, and so Yahoo took off points. If you didn't do it at all, you can complete that part. Just do use t-test to get p-values, email the completed assignment to me and CC Yahoo, and we'll give half points back on that part. Yeah, if you didn't do the p-value calculation, you can do it now. Email it to me and Yahoo, and we'll give you half points on it. Okay. So we're going to play with a new R script today. I want to introduce the new homework assignment, and we'll play with some R code that's specific to Nextgen Sequencing data analysis. And if we have time, when we get to Nextgen Sequencing data, I said last time that the data are count data. They are read counts. We get these short little genomic chunks, and we sequence each one of them, get the pile of reads, and then we align each of those reads back to, usually align them back to a reference genome. And that allows us to say, for gene 1 and gene 2, and so on, if we have these little reads that here's one that got aligned to that location of the reference genome, here's another one, another one, another one, another one, another one. So we're going to, in our M by N matrix of numbers, when we're doing next-gen sequencing, those numbers are going to be counts. How many of those short little sequences did we align to gene 1? So if we counted all those up, that's what would go into capital X for gene 1. If this is mouse 1, say, row 1, column 1 would be the number of reads that overlapped at gene 1. Okay, so the numbers in our data set are counts. And that ends up being kind of important statistically, because if we have counts, let's say we want to compare gene expression levels between treatment and control. We could consider doing a t-test. I have numbers in treatment, I have numbers in control for gene 1. Let's do a t-test comparing those two groups. The t-test assumes that the data that you're seeing are normally distributed. At least when the sample size is small, it's a formal assumption that's being made. And with count data, your data are not normally distributed. Count data you could think of more along the lines of binomial data or Poisson data. So Poisson distribution is a probability distribution that applies to counts, like 911 call center in Houston, Texas. How many calls come in over the weekend? We're going to watch and count up how many calls come in. You might model that with a Poisson distribution. Binomial talks about a number of successes out of a number of independent trials, where each trial can be success or failure. So we could think of these NGS data as, here's my huge pile of reads that I haven't aligned yet. So I've got millions of reads. And I'm going to pick those up and try to map them back to the full genome. And each one of them is either going to map to gene 1 or not, yes or no. So we could say the count at gene 1 is like a binomial random variable as well. So what I'll show you either today or next time is that most of the methods for doing statistics with next-gen sequencing data are not t-tests, not linear regression, but they are just like those, but specific to count data. The reason you care about that, why does it make a difference? Should I really care? Why not just do a t-test? If you're going to bother to do statistics, you want at the end of your analysis for your statements to be interpretable and mean what you claim they mean. So if I have an estimated false discovery rate of 0.05, and here's my list of interesting genes, I need to have computed the p-values correctly for that FDR to be correct. So if I did t-tests with count data, chances are the p-values may not be right. I may get wrong inference. It'll probably be consistent, a similar story, but they won't be the right numbers. And what statistics allows you to do is be precise about how confident you are in your results. FDR estimate is 5%. That's my confidence level, kind of. And if I computed the p-values wrong, it might be 10%, or it might be 0.01. I don't know, because the methods I used didn't, I can't tell you how they work in this situation. All right. Okay, so over here, ngs.r. Let's open that up. Before I do that, let's look at the homework assignment. So homework 3, homework 4. So there's 2 questions, and these are both to do with next-gen sequencing data. The first question is a simulation that you're going to do in R. So we talked a little bit about simulation as a generic technique for exploring, what a statistical method's doing. If you can simulate a situation, you can often understand it well, because like Mitchell's doing, he's trying to simulate generations based on their parentage and their genetics and everything. If he can simulate the kind of data that you're going to observe, then you can play with the different inputs. What if I change this parameter about the data and things like that? What is the impact? Simulation is a good technique or a good skill to have. R is good to know, partially because it makes simulation pretty easy. You can simulate, you can program in R. Once you're up and running with it, you can do simulations pretty straightforwardly. So number 1 is basically a simulation where I'm going to ask you to simulate a genome and simulate this next-gen sequencing process. Let's talk through it. So we're going to say there's a genome that consists of 1,000 bases, 1,000 A's, C's, T's, and G's. That's how long the entire genome is for this imaginary organism that we're going to play with. Suppose that the genome is organized into 20 genes. Each gene is 50 bases long. So this is totally make-believe, but the entire genome codes for something. You can break it up into gene 1, gene 2, ... No introns, exons, anything like that. So 20 times 50 is 1,000. The entire genome is involved in coding for stuff. First 50 bases is gene 1, the next 50 is gene 2, and so on. Okay. Suppose that, okay, so this is a genome that would be in the cells of whatever we're studying. Might be a mouse, might be a rabbit. If we took a tissue sample from a mouse and extracted the DNA material, suppose we wanted to quantify the expression level for the genes in this organism. One way that we know to quantify expression level is to quantify numbers of RNA copies, in particular mRNA copies. These are just the intermediary copies of the gene, and for every one of them, ideally, there will be a protein. So if there's lots of RNA copies of a gene, it's highly expressed. Suppose we wanted to quantify and compare expression levels on the basis of next-gen sequencing data. This would be called RNA-seq data, because we're going to specifically use the little sequences, the little reads that we get to quantify RNA copies. Suppose that in a particular sample, like a mouse, we have a mouse and we extract a tissue sample. Suppose that in reality, in that tissue sample, there are 25 RNA copies of gene 5, there's 60 RNA copies of gene 10, and there's 40 copies of gene 15. And none of the other genes are currently being expressed. So in our actual data that we're going to run through this sequencing platform, this is what's really going on in the sample. There's no representatives of any genes except these 3. And for these 3, there's 25 copies of gene 5, 60 copies of gene 10, 40 of gene 15. So let's do sequencing. Let's do next-gen sequencing to obtain sequencing reads. And remember that when we do next-gen sequencing, we don't actually sequence the entire genome all at once. We break it up into a lot of little pieces. That's why we get these short reads. And depending on the specific technology you use, the length of those little pieces will vary. Some will give you shorter pieces, some will give you longer pieces. The longer the pieces are, you could kind of imagine, you get more information the longer the pieces of reads are that you get. It's easier to get short reads, it's harder to get long reads, but the longer the reads, the more information. We're going to pretend that we're using a technology that can read 10 bases. So even though each of these genes have 50 bases, we're going to be only able to read 10 at a time. So we're going to simulate that process. And then we're going to align our reads of length 10 back to the reference genome. We're going to make our own reference genome. You're going to simulate it in part A. And we're all going to simulate it exactly the same, so we all have the same reference genome. So then we will align our reads back to the genome. And then I want a picture. So the picture is intended to help you understand what next-gen sequencing is doing. This is the type of data that we're going to get from next-gen sequencing. So I'm calling this a dot plot, and I mean that to be the simplest kind of dot plot, which is just you put a dot above wherever a number corresponds to. You find that location on the x-axis and you put a dot above that location. If there were 5 realizations of the same number, you'd have 5 dots standing on top of each other. So you get this kind of piling up of observations. This is a realization of what you're going to do. We're imagining that we extracted a tissue sample. It contained X copies of gene 5, Y copies of gene 10, and so on. We did this sequencing. We could only sequence bases or fragments of length 10. Then we aligned back to the genome. Some of the time, we made an error when we aligned back. This is the entire genome. There were 1,000 bases. We said that only genes 5, 10, and 15 were being expressed. This is gene 5, I think, or maybe this is 14 reads base K in the genome. In this example, there were 2 expressed genes. So this is not exactly reflecting the setup in the problem, but you will have something like this. You have 3 locations where there's actually expression going on. Some of the time, when you get these little reads, you're going to align them back to the genome. You may not align them to the correct place. So these over here are incorrect alignments. These are reads that we pulled out of our simulated data. We threw them into that pairwise alignment function and put it in the wrong place. But over here, at these two genes, we have multiple reads that overlapped. So when we get our data matrix for this gene, whatever gene this is, with all these reads piled on top of it, the number for this mouse, say, and that gene is going to be the number of those little reads that are piled up here. A read count. So if you're familiar with next-gen sequencing data, you're familiar with the term read counts. Here's your read counts. That's what these are. They are counts of short little read fragments that mapped back to a specific location that we can say that's gene X. So I want you to simulate a genome. So you can use this code in part A. Then genome is going to be an object that is a bunch, it's a string, it's a character with lots of A's, C's, T's, and G's. It's going to be 1,000 A's, C's, T's, and G's. That's our genome. You are going to create the fragments that are going to be sequenced. Okay, so we are going to have 25 copies of whatever gene 5's sequence is, and 60 copies of whatever gene 10's sequence is, and so on. So you can create those in your R code. Here's 25 of this character string, here's 60 of that character string, here's 40 of that character string. Then we can't sequence the full length. We said that our technology can only sequence 10 bases at a time. So you're going to have to create fragments. Here's my RNA pieces. I'm going to break them into smaller pieces. That's what we do with next-gen sequencing. We take the genomic material, we chop it up into a lot of little pieces. So you're going to randomly pick a location in the transcript to break it. You're kind of pretending like you're an enzyme. You're going to randomly pick where's the enzyme going to chop this little representative of an RNA. So you pick that. If it is 10 or if it's less than 10 bases, throw it away. If it's 10 to 14, go ahead and sequence the first 10. If it's 15 or longer still, then fragment it again. So you're kind of simulating the process of taking RNA from a tissue sample, chopping it up into small pieces. That's what we've done so far. We've chopped our genomic material into small pieces. Now you're ready to actually sequence. So you know what the sequences are because you've generated the genome and then you randomly created copies, 25 copies of gene 1 and 60 of gene 10 and so on. But you're not going to write down the sequence that you know in that case. You're playing pretend. We're simulating. So you have to simulate. You have to sequence right now. You have the reads or you have the fragments, but you need to sequence them. So how you're going to simulate this sequencing is go ahead and take the actual sequence, but then allow for yourself to be wrong. Sequencing, we've said, is not deterministic. We have some level of confidence. Whenever I say this base right here is a C, I could be really confident that it was a C, or it could have been either a C or a G, so I'm really not sure. In which case, there will be some times that I make a mistake. I said it was a C, but it was really a G. I made a mistake. So allow there to be mistakes. And in particular, allow for sequencing errors to occur in 5% of all base calls. If you're going to sequence a fragment of length 10 bases, then basically you can flip a coin that comes up heads with 5% probability and say if it does come up heads, pick something other than the real base to put in there. Create a sequencing error. I will definitely help on the discussion boards with this particular problem. I think it's a really fun problem. It's a good problem. I think folks, it's a good problem that you learn through, but it is a fair amount of R programming, a little bit of programming here. So holler at me if you need help, and I will. You create a genome. You create the short little fragments of the RNA copies. Then you sequence those copies by allowing for there to be errors every once in a while. Then you align those reads back to the genome. So we have a pile of reads at that point. We have the pile of reads. Now we need to align back and line them up in the right place in the genome. You know how to do that because we did it in the beginning of the semester. Pairwise alignment. If you pass in, here's one sequence, and then here's another one that I just got from one of these reads. Here's a read of length 10, and here's a really long sequence of length 1000. Pairwise alignment. You can do that to every one of your fragments, every one of your reads. I tell you what parameters to use. Match reward of +1, mismatch penalty of -1, gap penalty of -10. Okay, do that, get a p-value for every one of them. Use the randomization-based test that we used earlier in the semester to quantify confidence level in the alignment. And only keep the reads that align confidently, so with p-values less than 0.05. Okay, then you're ready to actually create one of these things, a very simple version of one of these things with just one column and 3 rows. Because you can go count, you can tabulate how many reads. Let's see, what did I ask here? Oh, this is not exactly that. This is just going to tabulate how many times each base in the genome was covered at all. So any overlap at all between a read and a particular base. So at location 920, that's a C maybe, and there were 4 reads that were aligned that overlapped with that C. Then at that C, there's going to be 4 dots, 1, 2, 3, 4. There were 4 times that at this particular location there was a read that overlapped with that location. That's the information. Those counts are how you're going to make this picture, the read count picture. Statistically, what we're going to do is we're going to have a picture like this for mouse 1 in treatment group, and a picture like this for mouse 2 in treatment group, and so on. And then we're going to have pictures like these for all the mice in the control group. So we can go to gene whatever this is, gene 7 or something, and say the number for mouse 1 in treatment was however much that is, a few hundred or something. For mouse 2, it'll be a little bit different. Mouse 3, it'll be a little bit different. But I can do a test. Is the mean read count in treatment group different from the mean read count in control group? That's number 1. Number 2 is actual analysis of some RNA-seq data. RNA-seq, that's Nextgen Sequencing. It's using Nextgen Sequencing to quantify expression levels. We're going to tabulate, we're going to count up RNA copies, basically. Let's look at these data. I think we looked at these last time. They were, these are rats, right? These are rats, and each rat was exposed to either a treatment, which induced pain, or nothing. So treatment versus control. We also have measurements at 2 time points. So 2 weeks after treatment and 2 months after treatment. We looked at them. Let's go ahead and look at them one more time. Data, hammer. Remember, the pheno data shows you kind of the meta information. So there were 8 samples. The first sample was, oh, this is interesting. This seems to be saying these are technical replications, if I'm reading this right, that this is one observation of a rat in the control group at time 2 months. And this may be another observation of the same rat. I would need to read the paper to know for sure. But the fact that they're using this technical replicates here makes me think that this is just 2 runs of the same mouse. And we know something about issues that come up with doing that. If I wanted to do a t-test, you know, here's 6 numbers in treatment, here's 6 numbers in control. Is there a difference between treatment and control? I could do a t-test. But what if those 6 numbers in treatment are just 2 people that we measured 3 times each? And in control, there's 2 people that we measured 3 times each. Then I don't really have 6 observations and 6 observations. I don't have 6 independent observations. So if I were to do a t-test in that case, here's 6 observations, there's 6 observations, my inference would be incorrect, unreliable, because the p-value would be wrong. It would be treating it as if we have a sample size of 6, but it's more like we have a sample size of 2. So this is an issue. Maybe we can think more about it as we go. But for now, let's just say we have 2 control rats at 2 months, and we have 2 treated rats at 2 months. Then we have 2 control rats at 2 weeks, 2 control rats, or 2 treatment rats at 2 weeks. Here's the actual data, the read counts, M by N matrix. First column is the gene identifiers, and then we have a separate column for each of the rats, and then these numbers are counts. There were 2 short little sequences, 2 little reads in rat number 1 that were mapped back to the mouse genome and overlapped with the location that's chalked up to that gene. So the read count for that gene in mouse 1 is 2. The read count for that gene in mouse 2 is 4, and so on. And you can see there's lots of zeros. There are lots of zeros. There's some genes where there are non-zero numbers, but then zeros. That means that there were not any reads in that mouse that were aligned to that gene. There are other genes that have a lot. There's a lot of expression, high expression. So there's a lot of variability in what these genes are doing. Okay, so you are going to use this thing called DESeq2. I'm going to show you 2.5 methods that you can use for RNA-seq analysis. These are pretty widely used for RNA-seq. DESeq and something called EDGE-R and something called Lemma. Maybe you've heard of one or more of those. DESeq2, I think, is my favorite of the 3. So that's the one I'm going to have you use here. There's an R bioconductor package that does this for you, DESeq2. And basically, it's an entire package that's designed to work with read counts, tests and things like that specific to read counts. It has its own normalization algorithm that might as well use. It's built into DESeq. So instead of log transforming your intensity numbers, which you might often do in intensity-based assays like a microarray, intensities are often right skewed. They can't be negative, but they can be really big. So you might log transform right skewed data to make them more bell-shaped and think happier thoughts about using a t-test. Because we have count data, because of the unique nature of these data, there's a unique algorithm that's proposed for kind of normalizing or standardizing. Go ahead and do that. Once you've done that, do hierarchical clustering and do PCA. What do you see? Then do a test, gene by gene. So in this one, we need this special function from DESeq to do this. So I want to test something. I want to go, let's say this is gene I. Let's say YIK is the read count for gene K, let's say gene K, in sample I. Read count for gene K, sample I. And I'm going to model that with a regression model. I'm going to say the mean read count for gene K in sample I is an intercept plus a time effect. So let's say that time is a categorical variable. It could be, let's say, 0 for 2 weeks and 1 for 2 months. Time is 0, 1. Treatment is similarly a 0, 1 variable, a categorical variable that indicates was it the treatment group or the control group. And let's allow for an interaction. So let's allow for the treatment effect to be different at the different times. And that's sensible given the design of the experiment. You're going to induce chronic pain in these rats, and then 2 weeks after that treatment that induced chronic pain, you're going to assay their genetic profile. Then you're going to do it 2 months after that. So do you think that possibly the treatment effect would be one way at 2 weeks and possibly different at 2 months? Well, suppose that the treatment induces a response, but then you recover. The rat recovers or adjusts or compensates or something. I don't know. It doesn't take much imagination to see that at least possibly there could be an interaction. So if there's possibly an interaction, it makes sense to allow for an interaction. So here's a model that allows for a time effect, a treatment effect, and an interaction. And then I want you to specifically test. So usually we have thousands of genes, thousands of features, and we want to pull out the subset of them that we think is interesting somehow. And we will usually do that with a test, a hypothesis test, t-tests or linear regression, or a model like this one. So in order to filter out subset of interesting genes, go get some p-values. And the p-values I want are specific to testing whether there's a treatment effect. Is there a treatment effect? And we've seen this before once in our class. With a model like this that has an interaction, is there a treatment effect? What I want to do really is ask, can I pull treatment out of the model entirely? For example, as an alternative, you might say, why not just test whether beta 2k is equal to 0? Because when you fit a model like this in R or any other software package, you're going to get estimates. Beta 0 hat is such and such, beta 1 hat is so and so, and dot, dot, dot. You're going to get standard errors for each of them, and you're going to get a p-value. That p-value is applicable to the null hypothesis that this is equal to 0. Okay, so I don't want to go through this again. If you need tips on where this interpretation comes from, shoot me an email and I'll clarify it that way. But with an interaction, to ask, is there a treatment effect? We have the model allowing for a treatment effect that's specific to treatment group and a treatment effect that's specific to control group. So long story short, what I want you to do is test whether beta 2k and beta 3k are equal to 0. If those are both equal to 0, then I might as well pull treatment out of the model, because that's everywhere that treatment is. So I want you to test, is there a treatment effect? By asking, can I pull treatment out of the model? That corresponds to this null hypothesis that beta 2 and beta 3 are simultaneously 0. You can't do that from the output of R, the default output from R, estimates and standard errors. If you took 645 with me, you know a technique to testing a hypothesis like this is a likelihood ratio test or an F-test. You can do a likelihood ratio test with DE-seq2. And it's easy to do. You just have to fit 2 models. So we're going to use this in binom LRT to fit 2 models. One is going to be the model we started with. This is the one that I think is sensible for representing these data. This is the model that would hold under the null hypothesis that I'm considering. So this model down here is the same as the one above, except that we just pulled treatment out. So in the second model, we've forced the null to be true. There's an unrestricted model and there's a restricted model where it's been restricted so that the null is true. You fit those two models, you do a likelihood ratio test between them using this function, and it will give you a p-value. So if you go to row 1, gene 1, you can fit this model to gene 1, and you can fit that model to gene 1. You pass those fits into this function, and it will give you a p-value for gene 1. Is there a treatment effect? Show me the histogram of p-values, and this will also give you FDR estimates, like q-values. So the last couple are just manipulations of those results. Questions? Okay. I am going to ngs.r. I'm going to skip some of this. At the very beginning, there's some information about obtaining read data. So there are databases online, just like there's databases for downloading sequence information for a given gene. There's databases online that if you want read data for human beings, say, you can go download piles of read data. Or if you want read data for the fruit fly, you can go get piles of example read data. So that's what this first chunk is about in ngs.r. I'm going to skip this in the interest of time and get to a couple of things. One, I did want to mention this idea of a FASTQ file. We have seen a FASTA file in class, text file with a separate entry for sequences. FASTQ file is a similar idea, text file with separate entries for things. But the things that are going to be in the FASTQ are information on the sequences that were called. So we are going to have a name for each sequence, the actual sequence that was assigned. Hold on, is that right? Yeah, the sequence that is assigned. And we're going to have, interesting, a confidence score for each of the bases. So we have let's pretend we're doing RNA-seq. We have these transcript RNA copies. We chop them up into small pieces. We get short little reads that have been sequenced. And there may be errors in those sequence calls, those base calls. In a FASTQ file, we will have, for every single base in a read, so here's read number 4,000,002 is these bases from your mouse. For each of those bases in that read, we're going to have a number. Well, it'll actually be a character, and that character can be translated into a confidence score. How confident are you that this particular C is actually a C? I don't want to go into details too much, but those confidence scores are called Phred scores. They are, yeah, read this if you're interested. It's in the FASTQ file. And just bear in mind that when you're doing next-gen sequencing stuff, the read counts that you get, so our Excel spreadsheet of read counts, like in the hammer.txt file, whenever it says 2, that says there were 2 reads that were aligned to overlap with that gene. There's multiple ways that that could be misleading. One way is that the read that we actually obtained, one of these that we obtained, we could have missequenced it. There could be sequencing errors in it. If there's sequencing errors in one of these two reads, then it's possible that we misaligned it. If it's the wrong sequence, then how confident are we that it really goes with this gene? So just be aware that underlying these numbers, there is some uncertainty. There's some potential for misleading numbers. Hopefully, whoever gave you your data imposed some strict rules before they populated their data set. And one way you can do that is with stuff like this Phred score. So I'm only going to keep reads, for example. I'm only going to import or increment counts in this table for reads that I'm really confident about. I am going down here. Line 142, I want to at least comment on two standard file formats in the context of next-gen sequencing data. You obtain your tissue sample, you extract your genomic material, you chop it up into small pieces, you run it through the sequencer to get short little reads. The FASTQ file is a place where we can put all the reads. But all the FASTQ file has is the sequences of the little reads and confidence measures for the sequences themselves. But the FASTQ file does not tell me where in the genome each of the reads lined up with. So just with the FASTQ file, I'm not ready to draw one of these things. I don't have the information yet that allows me to say where each of the reads lines up. So I'm not ready yet to create my capital X matrix of read counts. The next piece of information that I want is to do with alignment. Where do I line each of these reads up? And there's a standardized format for passing that information around. There are SAM and BAM files they contain the same information, just in different format. The BAM file is a binary version of a SAM file, basically. And they have to do with alignment. Okay? So when we obtain our reads, we've talked about wanting to align them back to a reference genome. So if we're studying rabbits, we might take a pile of reads from tissue samples of 10 rabbits, but then we need to map those reads back to the reference genome for the rabbit. All right. Alignment is not typically something that you would do in R. Just FYI, if you're doing NGS data, and all you have is a FASTQ file, I don't think I would recommend trying to pull off a large-scale alignment in R. I don't even know if there's functions that try to do it. There's usually standalone packages, programs for doing alignment, because it's very computationally intensive. Because you're going to have millions of reads and you're going to have millions of bases typically in your reference genome. So it's lots and lots of calculations that have to be done. But once you have done the alignment, what the widely used platforms will give you, like Bowtie, for example, is the data, the alignment data, in a specific format, a SAM file, or the binary version of the SAM file called a BAM file. So the SAM files contain the sequence for each read and the location in the genome to which it aligned. So that's going to include, that might include like chromosome 4, starting at base number such and such, length so and so. If you tell me that, I know exactly where in the genome to line up, which means I know exactly where in the genome to increment, count, one extra count goes with that gene. One extra count goes with a gene if this read aligned with that gene. You can manipulate SAM and BAM files in R. So if you do NGS and you use something like Bowtie to do your alignment, you can extract the SAM/BAM file from Bowtie and pull that into R and go from there. R can work with BAM and SAM files. So here is some examples if you're interested, if you want to dig into SAM/BAM files. There's an R/SAM tools package on Bioconductor, and it will read in BAM files. I think it works with SAM files too, but BAM is binary, so it's a little neater to work with for the computer. So you can pull out of the SAM BAM file specific reads. You can pull out for a given read, like where it aligns, what the alignment information is. You can pull out the sequence. You can get pretty much anything you need in order to create one of these. All right. What I wanted to spend some more time on in class together is what comes next. So starting at line 187, I have some content on 3 widely used methods for next-gen sequencing. So widely used statistical methods for next-gen sequencing data, specifically RNA-seq, which is the most widely used, I think. One is called EDGE-R. One is called DE-seq2. There was a DE-seq original one. Now it's been revised to DE-seq2. And less ideal is called Lemma. Lemma was actually developed in the context of gene expression microarrays. And we said t-tests and stuff that we usually use for microarrays aren't ideal for next-gen sequencing because we have read counts. It's count data. So what Lemma does is basically transform your counts in a fancy way to make them like continuous data, and then just do all the microarray stuff. So Lemma is one possibility. I'm going to emphasize DE-seq2. Based on what I've read and seen, I think maybe it has an edge over the other two. Both DE-seq2 and EDGE-R are very similar. They just differ a little bit in the normalization algorithms they use and a couple of other things. But both of them are basically, let's do statistics on count data. One of the details, we're about to run out of time, but one of the details with our count data in next-gen sequencing is there's what's called over-dispersion. Let me show you a picture real quick. We'll end with this. Because if you do next-gen sequencing, you know, I've mentioned this a couple of times, you're not doing t-tests anymore. T-tests, most people know what a t-test is, or a simple linear regression model. If you go read a paper about next-gen sequencing, RNA-seq or something, they'll be talking about negative binomial regression and stuff like that. Some things that aren't what is taught in STAT101, for example. Where does that come from? Why is it more complicated all of a sudden that we're doing next-gen sequencing? Let me just show you the issue. I'm going to simulate some data. Sample of size 1,000, and I'm going to generate some Poisson data. So let's look at what Poisson data looks like. Sample size 1,000. Poisson is a distribution applicable to counts. A Poisson random variable can be 0, it can be 1, it can be 2, ... all the way up to infinity. That's what Poisson's for. So let's generate y_p, some Poisson random sample of 1,000 draws from the Poisson distribution with mean 10. For a Poisson distribution, a Poisson random variable, the mean is equal, in this case we're calling it mu, that's 10. Who knows what the variance of that variable is from STAT 101, 102? What's the variance of a Poisson random variable? If the mean is equal to mu, what's the variance equal to? Remember? It's equal to the mean. Right. It is the mean. So the variance should be equal to the mean. It won't be exactly equal in this case because it's sample data, but the population mean and the population variance are exactly the same if it's Poisson. These are going to be pretty similar. They're pretty close. So a technique that assumes the data were Poisson would be fine. There's a way to do regression, for example, with Poisson data. And we have no problems here because our data are Poisson. When we do read counts, though, next-gen sequencing, it is not Poisson because of this mean variance relationship thing. So let me do this. I'm going to generate some more data, and these are going to be generated from a different distribution. It's called negative binomial. And if the first time you learn negative binomial, you won't see the connection to this Poisson stuff. I'll show you the connection next time. Basically, when we do next-gen sequencing, we're getting counts, but it turns out that they have greater variance than the Poisson distribution would predict. Their variance is bigger than their mean. When that happens, it turns out that a negative binomial distribution can model that well. So negative binomial, I'll show you why next time. Think of it as Poisson, where you've allowed for extra variability. So here are negative binomial data, and let me show you the mean and variance of these two things. So the mean and variance of our Poisson data were Poisson-like. The mean and variance of data that I generated according to a mechanism that's like what we're going to get with next-gen sequencing, mean and variance are nowhere near alike. So whenever I can't do something like Poisson regression here, unless you have to make it a little more sophisticated to allow for this. And that's basically what all we're doing. With EdgeR and DESeq2, we are doing Poisson regression, where we allow the variances to not equal the mean. So you might hear that referred to as over-dispersion. If you try to read a paper that talks about EdgeR or DESeq, you might hear of over-dispersion, which is why that's where this negative binomial idea comes from. So I've got some comments in the text file. One of the challenges with using a binomial or a Poisson model for NGS count data is that the counts are over-dispersed. The variances are bigger than what your model would say if you picked these two models, one of these two models. So a common solution is to use Poisson, because it makes sense, it's counts, but just make it a little more sophisticated to allow for the variance to depend on something else, another parameter. So instead of just one mean parameter that's mean and variance, we're going to say there's the mean and then there's a scale parameter as well. Turns out that is negative binomial. So if you say there's a Poisson, and for the Poisson rate parameter, if you give that a distribution, then the marginal distribution is negative binomial. I'll show you that next time. Long story short, we're going to use EdgeR. Actually, mostly going to use DESeq2. They both do virtually the same thing. They are going to use this negative binomial model to do tests and give us p-values and FDRs for genes. But it's based on next-gen sequencing read counts. What follows after this stuff, I won't spend too much time on it. It's, for example, a way that you can do gene ontology enrichment with RNA-seq data, or KEGG enrichment with RNA-seq data. There's also a package that's specific to DNA methylation analysis, which is an epigenetic mechanism, and a couple of other things. But there's also ChIP-seq. I will talk a little bit about ChIP-seq. So 2 or 3 more lectures on next-gen sequencing, and then we'll move to the next thing. All right, are there questions about that stuff? You can get started on the homework if you want over the break, tinker with it a little bit. I do recommend that you get started sooner rather than later on the simulation, because that, I think, is something that you will process after thinking about it in a couple of sittings. Okay, I'm done. Have a good weekend and a good break.